welcome to the channel. And so previously we were studying the differences of the germination of the seeds. Now today we are going to learn about the germination, the factors affecting the germination. So the germination, thank you. Bye. So the germination is nothing but the growth of the seed to an embryo, that is a small baby plant. So it is, this process is known as germination. And there is also something called dormancy. This is the phenomena called dormancy. In this particular content, dormancy means when the seed does not get suitable conditions to grow, then it remains as it is until it receives that suitable condition. So this phenomena is known as dormancy. Okay, now there are factors affecting germination. The factors affecting germination are water, optimum temperature is required, water is required, and air is required for the respiration or oxidation. Okay, so in the types of germination, in water, uh, the water helps in the following ways. So when the seed absorbs water through micropyle, then becomes it starts to become big. It it starts to become big, and then the seed coat of the seed bursts. Um, it bursts, and then the radical comes out of it. So water is used. Water is helpful for the radical to come out of this seed coat, and then water uh, water also is helpful in the transportation of soluble food from the storage tissue to the developing embryo. So the water also acts as a transport agent from the storage tissue to the developing embryo. So it transfers food to the developing embryo from the storage. And also it is the essential medium for all the chemical reactions that takes place. The stored food converts into a more soluble form and is used by the embryo for its growth. Now these are the only 3 points which you need to learn. And also we will be learning about oxygen and temperature. So oxygen here is, what are the uses of oxygen for the plants? So when the plants absorb oxygen, it is important for oxidation and respiration. Oxidation of the food and respiration is required for the energy. So during germination of seeds, metabolic activities accelerate, and rapid cell division and cell growth follow, requiring energy. This energy is released from the stored food materials by the process of respiration or oxidation of food. For this purpose, the seed require oxygen. Temperature. Temperature plays an important role. So there should be an optimum temperature for the seed to grow into an embryo. Okay, embryo is a small baby seedling, you can say. So a seed to seedling, we require water, oxygen, and optimum temperature. Without these three factors, the seed cannot go and it achieves the state called dormancy. Is that clear to everyone? Okay. So optimum temperature. Why do you call it optimum? Optimum is something that is suitable. Something that is suitable for the plant. So optimum for the plant. So optimum temperature is the suitable temperature. The temperature varies. It cannot be constant for the plants of all over the world because there are different horizons. There are different temperatures in Arctic pole, in Antarctic pole. There are different temperatures, and in Sahara desert there are different temperatures. Similarly, here we are talking about temperature, which plays an important role in the germination of seed. If it is very hot, then it will damage the tissues, which will affect the growth of the embryo. And also, if the temperature is very low, then the metabolic activities cannot accelerate, and then it will remain as it is. Now there is process of seed germination. So what happens in the process of seed germination is that there is a seed. You have dig the mud and then insert the seed. Water the plants. Water is required. So when water is poured, then the seed absorbs water. When the seed absorbs water through the micropyle, then it starts growing bigger and bigger. The seed coat, which is the protective covering of the seed, it ruptures, and then from there the radicle comes out, which later turns out to be the root system. Okay, and then after the radical comes out, the temperature also has an important role in the germination of seed. So temperature is required, and also the water absorbs the root absorbs the water through diffusion. So and then it passes all over the plant body. So like that, the plumule also grows out, um, and it forms the shoot system. Okay. Then between this process, the embryo produces a large number of enzymes that hydrolyzes the food material and make it available to the growing embryo. With the availability of food, cell division starts in the embryo. Radicle and plumule grow in size and emerge out of the seed coat. The radicle is the first to come out of the seed coat. It forms the root system, then grows into the soil. Then the plumule comes out because when we water, then the water enters the root through diffusion processes, osmosis, diffusion, and all the stuff. We have already learned about diffusion and osmosis. I will explain it to you in next video, particularly on diffusion and osmosis. Okay, I will teach that in detail. That will be helpful for the further studies. Okay. And then the germination of seeds requires a sufficient quantity of water, air, and optimum temperature. So now they have given here the experiments to prove that seed actually needs water, it actually needs air, it actually needs the optimum temperature. So for that, In the experiment 1, the materials required is the conical flask, the petri dishes. So 4 petri dishes, let's name them A, B, C and D. So I am writing those 4 petri dishes here. So petri dish A, petri dish B, petri dish C and petri dish D. Okay? Just a second everyone. So there are 4 petri dishes. Petri dish A, petri dish B, C and D. We will keep equal seeds in the 4 of the petri dishes. So 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4 and 1, 2, 3, 4. Now these are the seeds I have taken. Now we need the cotton wool or blotting paper. Here I am going to take the cotton wool. Okay, so here I am going to take the cotton wool. You can take cotton wool or blotting paper. Okay, and in petri dish A, the cotton will be moist. Okay, in petri dish A, the cotton will be moist. In petri dish B, the cotton will not be moist. Okay, will not be moist. In petri dish C, the cotton again will not be moist. And then in the last petri dish, the whole seed will be submerged inside the water. Okay, so in petri dish D, the seeds are completely submerged inside the water. Okay, now this seeds here, the seeds will be soaked overnight. This also will be soaked overnight. This will not be soaked, and this is submerged. Okay, so now I'll explain what I have written here. So there is petri dish A, B, C and D. So there are equal number of seeds that I have given 4. So we have used the materials like 4 petri dish, cotton wool or blotting paper, and then some seeds you can take gram seed or bean seed or any seed. Okay, and then you need to place the seeds on the cotton wool or blotting paper. Is that clear? Now in petri dish A, what they have done in petri dish A is that they have moistened the cotton and the seeds were soaked overnight. Okay, and in petri dish B, there's cotton is not moist and the seeds are soaked overnight, but the cotton is not moist. Now in petri dish C, both the cotton is dry and the seeds are unsoaked. Okay, and then in D, it is fully submerged inside the water. The seeds are fully submerged inside the water. Okay, now we will leave this for to 3 days. How many days? 2 to 3 days. So when we leave it for 2 to 3 days, we need to make some observations. Okay. So in petri dish A, what observation can we make? That the seed has germinated properly. Okay. And B, it starts to germinate, but then uh, it stops. Okay, it stops in between. I'll tell you the reason why. In petri dish C, as the seeds are unsoaked and the cotton wool is dry, so no change in petri dish C. In petri dish D, the seed may start to germinate, but then they will eventually rot because will be in the inference. Okay, so in inference they will give the reasons why those things which we observed happened. Okay, so let's read that out. In inference, proper germination of seeds needs adequate quantity of water, as in petri dish A. In the absence of water, seeds do not germinate, as in petri dish B. Deficient and excessive water also does not allow the germination to continue if O2 is not available. Once the O2 dissolved in water is observed, after that what? They won't have oxygen, right? So without oxygen also the seeds won't grow. So this experiment was to show you guys that adequate water is necessary for the germination of seed as well. Okay, now the next experiment, we need 3 petri dishes. Okay, so in experiment number 2, we need 3 petri dishes. 1, 2, 3. Okay, there are 3 petri dishes. We will do their namkaran. That is A, B and C. Okay, now the same procedure. We need cotton wool here, here, here. We need same number of seeds, same number of seeds, same number of seeds. But now it's not about water. Now here it is about the temperature. It is about the optimum temperature which it receives. Okay, so now you guys may not be able to see this. Okay, so in petri dish A, we will keep the petri dish A in the room temperature itself. Okay, room temperature. In petri dish B, we will keep it in freezer. That took for 2 to 3 days. Imagine, guys. And then this petri dish C will keep in oven for like 48 degrees Celsius or more. What will happen if the seeds get more temperature? The tissues will okay. So the tissues will be spoiled. It will damage the tissue If the temperature is high and if the temperature is low, it will stop the metabolic process and eventually the germination also will stop. But in the room temperature, it is getting adequate amount of heat, so that's why it is growing. In the petri dish A, the observation is that the petri dish A germinates properly, petri dish B and C does not germinate. Why does it not germinate? I have told you. Okay. Now experiment number 3. First experiment was about water. Second about temperature. Now guess what will be the third one? The third factor which affects the germination of seed. The presence of oxygen. Correct? Okay. Now for this we need, what do we need? We need 2 conical flasks, cotton wool or blottian paper, the same thing, but this time we are using conical flasks and not petri dishes. So we need 2 conical flasks. We need sodium hydroxide, and then we need pyrogallic acid and thread, then two small test tubes, and then soaked seeds of gram. You guys need to mention the soaked word, okay? And then I will tell you one thing, that pyrogallic will react with NaOH, then only it can observe, when it reacts with NaOH, then only it can observe oxygen. Okay, we are using pyrogallic acid or alkaline pyrogallic acid for observation of oxygen, so that we can get to know that what happens if the seed does not get oxygen. Okay, so let's check. So the procedure goes like this. We take 2 conical flasks and label them as A and B. You can label it as anything you guys want. Okay, and then the moist cotton, in each flask some soaked grams of equal amount of soaked gram, and then hang a small test tube containing alkaline pyrogallic acid. Alkaline is nothing but, why do I use nothing but, never mind, pyrogallic acid plus NaOH. So when it reacts with any salt, then it becomes alkaline. So pyrogallic alkaline with NaOH, which is a salt. There are different types of salt. NaCl only is not salt. There are different types of salts which you will learn in 9th grade. In the language of chemistry only you guys will have learned, no? The first chapter in the chemistry, isn't it? Okay. Now we need 2 conical flasks. Okay, blah 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 blah. We need to hang the test tubes inside the conical flask, with one with pyrogallic acid, the mixture of pyrogallic acid and NaOH, and another with simple water. Okay, and then there we will put the stopper, the airtight stopper, so that the air does not enter inside it. Okay, so what is the observation you can see is that in A the seed does not germinate at all, but in B the seed germinates properly. Why does it germinate properly? Because there was oxygen in it. But in conical flask A, the oxygen was absorbed by the pyrogallic alkaline acid, So there was no oxygen at all for the seed to grow or for the seed to respire. So therefore the seeds died and they could not grow or they could not germinate. So this was the third experiment. Now the fourth experiment is the most famous one. It is called three bean experiment. Okay, what is it called? It is called three bean experiment. This is very popular among ninth graders at least. Maybe you guys haven't have heard or somewhere. Okay, so here, just a second. Okay, so where is the board? And yeah, so we'll just be needing a single beaker, a glass rod or a wooden stand, a slanting uh, wooden plate or glass slide. Okay, somewhat like this, and the beaker will be half filled with water. Okay, it will be half filled with water. Now, in the three bean experiment, we will attach the seeds in this following manner: one out of the water, one in between the water and the air, and one fully submerged. What do you think, which one will germinate? So in the observation, this, let's name it A, this as B, and this as C. Okay, so this as C. So what do you think, which one will be germinating? You guys can comment if you want. You can pause the video and comment. I'll check the answers if it is correct or not, or maybe I should improve my explaining skills or something like that. For now, I'm going to give you the answer. You guys can pause and give me the answer in the comment box. Okay, so the B1 will germinate, the C and D will not germinate. Why will it not germinate? The C. So the C1, it is fully inside the water. The dissolved oxygen it has absorbed. Now maybe it will start to germinate, but suddenly it will stop and then it will rot. Okay, so it stops and rots. Nice rhyming word, no? Okay, so this one is this. So then B germinates properly. Okay, C may start germinating, but it will eventually rot. B will germinate properly because it is in between. It is getting adequate amount of water and adequate amount of oxygen, so it is able to balance. And then please keep this beaker in the sunlight, okay, in the room temperature, okay, so that it gets adequate or optimum temperature. Okay, then why didn't the A germinate? Because it was out of water. It only received oxygen and optimum temperature. But what about water? Humans only cannot survive without water. How will these seeds survive without water? So therefore, the top seed A does not germinate. The middle seed B germinates properly. The bottom seed C starts germinating, but soon stops and then rots. as I said. Uh, Inference is okay, I have explained. Now, there are types of germination. No, mind, I'll write it here. So, there are types of germination. What are they? Let me tell you this. So, there is hypo. So there is hypogeal and epigeal germination. There are two types, mainly two types, hypogeal and epigeal. Did I not teach you guys about what is it? It was epicotyl and hypocotyl. Did I not teach that to you guys? The epicotyl, the hypocotyl, epicotyl between the mesocotyl and the plumule, hypocotyl between the mesocotyl and the radicle. So that's what it is. The epicotyl and the hypocotyl. Here there are two types of germination, the hypogeal germination and the epicotyl germination. Epigeal germination. So, the difference between epigeal and hypogeal germination is that in the hypogeal germination, the germination takes place below the ground. Cotyledons remain below the soil surface, but in epigeal germination, the cotyledons come out of the ground and the germination takes place above the ground. And then in the hypogeal germination, epicotyl is long, and in epigeal germination, hypocotyl is long. Hypogeal germination, epicotyl is long. Epicotyl germination, epigeal germination, hypocotyl is long. Okay. Now, mostly found in monocotyledon seeds is hypogeal germination is mostly found in monocot state. Epigeal germination mostly seen in dicot state. Okay, and then the examples are gram, maize, pea, mango, groundnut, etc. And the epigeal germination, it is bean, castor, papaya, cotton, sunflower, etc. Okay. Okay. Now we will learn about the viviparous germination. What is viviparous germination? Ow. Okay. Viviparous germination. Viviparous. So viviparous germination is the phenomena where the seed grows inside the fruit itself. It does not come out of the fruit, it does not fall in the ground, it does not, all that process does not happen, it does not take place at all. What happens is the seed grows inside the fruit itself, and I'll show you this part. Okay, so this is the fruit of rhizophora. Okay, fruit, radicle, hypocotyl. I hope you guys are able to see. Okay, so that was it. And the examples of viviparous germination is rhizophora, cereopes, Avicennia, Heritaria, Sonaceae, etc. There are unique names with unique mode of germination. So you guys have understood, I guess. So this lesson is completed. Thank you everyone. Take care. Read well. I have explained so far. You guys also keep hydrating yourself just like those seeds. Okay. Next lesson will be, I think, respiration in seeds, respiration in plants. So the next video we will do respiration in plants. Okay. For now, bye bye. Thank you. Like, subscribe for these types of videos. I mean, these types, sometimes it changes, like I may dance, I may sing, I may do a jogging video or anything like that. So if you guys are interested in these type of binge watching, then please welcome. Thank you.